Today I'm going to review the background information and procedure for the McMush Lab. The goal of the McMush Lab is to test a McDonald's Happy Meal to determine whether it contains proteins and or lipids and or simple sugars or monosaccharides and or starch. First things first, this is a wet chemistry lab. As such, you need to wear safety glasses. I'm already wearing mine, so I'm good to go. But before you start the lab, please be sure that you obtain and wear your safety glasses or goggles. So before the lab begins, I will have created what I call the McMush. Essentially, I add all of the components of a McDonald's Happy Meal to a blender and create a McMush slurry or puree. And the individual components of the Happy Meal include a hockey puck looking cheeseburger with white buns, processed cheese, pickles, ketchup, and some sort of a meat patty. There's also an adorable little container of McDonald's French fries that also goes in the blender without the container, obviously. The Happy Meals contain some token healthy product. In my case, it's a small little bag of apples. And then you can also order a Happy Meal with milk, either whole milk or skim. I actually chose the least healthy option, and I chose regular Sprite. All of that went into a blender, and from that I created a puree of McDonald's happiness in a beaker. It's actually really disgusting. This is not the McMush. I didn't have access to a Happy Meal while I was doing this video, but the actual McMush would be thick and kind of gray looking, and probably nothing that you would ever be tempted to eat once it was put into a blender. Now, to determine whether or not each of the macromolecules is present in the McDonald's Happy Meal, you essentially put a small sample of the McMush slurry into one of each of 3 test tubes, and then you add one of 3 chemical indicators. Now, these are chemicals which will turn color, or a color change will be induced if the molecule that they're designed to test for is present. So, for example, if I added this to the McMush and it tests for protein, and there was protein in some component of the McMush, this would turn color. The question is, what color should this change when the molecule it's supposed to test for is present? So what we need to determine that is some positive controls. In other words, we have to test each of these indicators with the substance they're designed to test for. Whatever color change we get then, we know what a positive test result will look like, and we can compare any color change we observe when we add these to the McMush. And if they're the same color, we can infer that that macromolecule is present. Confusingly, 2 of the color indicators or chemical indicators that we use are both color blue, and they're both named with the letter B. So it's nice and confusing, but if you follow your lab manual and or this video, hopefully it will unconfuzzle you. The first is something called Biurette's solution. Biurette's is kind of this nice light blue solution. And this will turn color or tests for the presence of protein. The second is, I'll go with this one. This is Lugol's iodide. It comes in a dark brown bottle. You can buy iodine, which is a close relative, chemically speaking, of this, and you can buy this at a pharmacy. And it will also come in a brown bottle because this is photoreactive or photosensitive, so it reacts in sunlight and probably breaks down, so we put it in brown bottles to preserve it. When you take it out with a dropper, you might see that it is kind of a dark reddish brown color. So that's the Lugol's iodine. This tests for starch. And last but not least, we have something called Benedict's solution. This is the other blue solution that starts with the letter B, and this tests for the presence of simple sugars or monosaccharides. The last test, which I'm not going to show you, is a lipids test, and we don't actually use color indicators for that. We simply use a piece of paper, specifically like a brown paper bag that's been cut up. And the idea is lipids and grease, when it wicks or is absorbed by paper, will cause the paper to become sort of that filmy, translucent. So if you've ever carried a paper bag with maybe some fast food in it, which has lipids, you'll notice these sort of little grease stains in the paper. They're not water, they don't evaporate, they're permanently causing the paper to be that sort of translucent color. So to test for lipids, you would just dip the brown paper into the McMush slurry, leave it on the desk to dry. Once the water's evaporated, if it's still greasy looking, then that's a positive test result for lipids. So I have done the positive controls already. We will have done these together in class, so you probably don't have to do them for your lab. But let me show you what the positive test results look like. This is the Biuret solution. This is what it started with. This is what a positive test result looks like. It's kind of purpley. I don't know if you can see the difference. Often putting it up to a piece of white paper maybe helps you see the test results a little bit more clearly. But it goes from a light blue to a light purple in the presence of protein. The Lugol's iodide test, actually the starch that I used as my positive control when I did this test has settled down to the bottom. Right, so the sort of reddish color that we see here is the Lugol's iodide, which is red or reddish brown. And the very bottom of this test tube contains a starch, which is kind of a dark blue or purple color. Now, if you put your thumb over the test tube and invert it a couple of times, that will mix all that up. And so now you can see perhaps that's a much darker bluish purple color. It's maybe a little hard to see on the video, but you'll see it in class. Last but not least, this nice orange color is a positive test result for monosaccharides or sugars. This is the Benedict solution, and the only secret here, or the trick with this solution, is once you've added the substance being tested and the Benedict solution, you have to put it into a beaker with boiling water. The heat is what induces the chemical change. So if simple sugars or monosaccharides are present, once this is boiled for, I don't know, 3 to 5 minutes, it will turn this orangey color. This has also been sitting for a while, so a lot of the orange sediment is in the bottom. So if I, again, put my thumb over it and invert it a few times to mix, you can perhaps see this nice orange color, which indicates a positive test result for simple sugars. Now, to actually do the lab, you need your 3 test tubes, some McMush, and then your 3 indicators. Be sure and record which one's which, what's going into what tube, what indicator is going into which test tube. And read your lab manual for the specific volumes you need to add. It's a pretty forgiving lab. If you're not exact, you can still get a good test result. Right? You'll still get color change. It might just be a little harder to see if you don't get the volumes just perfect. I think it's 3 milliliters of each solution. These graduated pipettes actually have little marks along them. This full pipette is 1 milliliter from here to here. So if you can suck up 1 milliliter and hold it in this part of the pipette, you will have exactly 1 milliliter. If you suck it up and just allow the bulb to suck all the liquid up, you may wind up pulling liquid up into the bulb of the pipette. Well, now you have more than 1 milliliter. I'll show you what I mean. When I suck it up like this and hold it, right, now I have 1 milliliter. If I do this and just allow the bulb to completely suck up all the liquid, well, now I have liquid which is up into the bulb of the pipette as well. And so now I have more than 1 milliliter. So it's a little bit of a trick. You pinch the bulb, you hold it, you suck up 1 milliliter into the barrel of the pipette, and then you hold the pipette, still squeeze it, and you can maybe see I have 1 milliliter. I'm not going to do this for all 3, but you basically add 3 milliliters to one of the tubes, and then repeat for the other 2 tubes. Then, again, following the instructions in the lab manual, you simply add whatever it is that you're testing, each of the different indicators. The starch test and the protein test will be immediate. Once you add the indicators, if the molecule you're testing for is present, the color change should happen instantly. Remember, though, that the Benedict's test for simple sugars, you will not see a color change even if sugar is present until you put it in the boiling hot water bath for anywhere from 3 to 5 minutes. Once you're done, each of the tests, you can compare them to the positive controls to determine whether or not the molecule you're testing for is present. Then you can pour these down the drain and wash out the test tubes. I'd like it if you could use a little bit of soap and water and a small bottle brush that I'll provide in your lab kits. Once you've washed out all the test tubes, you can invert them upside down in the test tube rack to let them dry. Replace or return all of the indicators back to the basket at your lab station. You can pour this down the drain, the McMush, or in the trash if it's super thick so it doesn't clog my drain. Rinse out your beaker. You can throw away these plastic pipettes. I can't reuse them. And once your lab station is entirely clean, I would suggest that you wash your hands, especially if you used your thumb to invert the test tubes. Either way, I'd still say it's good lab practice to wash your hands after lab, and then you're ready to work on the lab write-up. The Benedict's test is a test for simple sugars, and this is the only test that you need to boil in order to generate the chemical reaction that causes the indicator to change color. So in order to do this, you need to add about 5 milliliters of the McMush solution to a test tube, and then about 3 milliliters of the Benedict solution, and mix them up a little bit, and then bring this test tube over to the hot water bath. Now, obviously, because we've got boiling water, the beaker and the water are obviously hot, but also the ceramic surface of the hot plate is exceedingly hot. So you have to be very careful, both because of the splashing boiling water, but also because the ceramic surface of the hot plate is white and it doesn't look hot, but it's the surface that's heating the water. So it is. Now, to put the beaker in the solution, if you're careful, you can just kind of put it in to the boiling water in the beaker like this. Just gently lower it in. Try not to drop it in. That might cause the test tube to break on the bottom of the beaker. And then you need to leave it. Now, it's the old adage of the watch pot never boils. Well, the boiling Benedict solution never changes color, at least when you're watching it. Nevertheless, the reaction takes about 2 to 3 minutes. If there actually is sugar present, then it should turn an orange color. And if there is no sugar present, it will stay kind of a blue color. Now, I just have a test tube full of water here, so there's not going to be any chemical reaction. I just did this to show you how to do the test. But after anywhere from 3 to 5 minutes, if your test tube sort of looks a little bit like this, which is kind of a yellowy orange color, right? If that's now in the hot water bath and it's an orange color, you can pull it out in one of 2 ways. You can use the test tube clamps, but they're not really great at gripping the test tube. So the best thing to do if you're going to use the test tube clamps is to grab the test tube, pull it out, and immediately pull it or put it into a test tube rack. Do not carry the test tube around the classroom with the test tube clamps. They almost always will drop out, right? So though you just kind of pinch the clamps together and you pull the test tube out, and it's already starting to slip a little bit, you can see. So maybe that's just to get it started. The top of the test tube should not be all that hot. So you can gently touch it with your fingers, and this has been in there for a couple of minutes, and it's actually safe to handle if you're careful. Obviously, you don't want to reach down and grab it from the bottom. The other way is, if you're careful, again, just avoiding the steam and boiling water very carefully, you can just grab it by the very top of the test tube and, again, quickly lower it down into your test tube rack. You do not want to carry the test tube around the classroom, either with a test tube clamp or with your hand.